Hi everyone, my name is Junyan. Today I'm going to be talking about my thesis, which is titled Multi-omic Systems Level Approach to Investigate the Immunometabolic Role of FAT10 in Cancer. My research revolves around the protein of interest known as FAT10. FAT10 is an abbreviation for HLA-F adjacent transcript 10. This name alludes to the location of the gene which is close to the HLA-F locus on chromosome 6. It is a ubiquitin-like protein, and like ubiquitin, it can interact with hundreds of proteins in the cell. FAT10 expression is inducible by the pro-inflammatory cytokines TNF-alpha and interferon gamma. It is important to study this protein because it is an oncogene. It is highly overexpressed in inflammation-associated cancers such as that of the GI tract. Across different studies, FAT10 expression has also been associated with poorer patient clinical pathological characteristics as indicated in red. FAT10 overexpression in vitro and in vivo also promoted a cancer phenotype. Several mechanisms have emerged to explain FAT10's oncogenic properties, and they revolve around FAT10's ability to interact directly with many proteins in the cell to induce one of three effects, namely destabilization, stabilization, or delocalization. For the purposes of my research, I'll focus on destabilizing and stabilizing effects. For destabilization, FAT10 can interact with many proteins and target them for proteasomal degradation like ubiquitin. FAT10 destabilizes a known tumor suppressor, WISP1, in the context of cancer. Interestingly, FAT10 can also conversely stabilize proteins by antagonizing their ubiquitin-mediated degradation. It does so for multiple oncogenes like beta-catenin, YAP1, and EGFR. Interestingly, apart from being a link between inflammation and cancer, FAT10 also seems to be a link between inflammation and metabolism from two separate studies. In the first study, when FAT10 was knocked out from mice, these mice lived 20% longer and were much leaner, suggesting that FAT10 promotes aging and obesity. Congruent with this, the second study showed that FAT10 promotes adipogenesis and lipogenesis in porcine adipocytes. This brings us to the gaps in knowledge which my study hopes to address. Firstly, FAT10 appears to induce metabolic changes in a physiological context, but whether or not it does so in the pathophysiological context of cancer has not been explored. Secondly, there's currently a fragmented view of FAT10's role in cancer. FAT10 can interact with hundreds of proteins in the cell, but thus far, studies have focused on functional characterization of FAT10's effects on individual interacting targets. This leaves us to the question, is there an overarching theme of FAT10's effect in cancer? Therefore, my study aims to integrate proteomic, interactomic, and metabolomic profiles in a cancer cell model to gain a system-level understanding of FAT10's role in cancer, and I focus on FAT10's stabilizing and destabilizing effects. I hypothesize that FAT10 modulates metabolism-related pathways of cancer cells by altering the proteome through stabilizing and destabilizing effects. My methodology is as follows. I first generated a colorectal carcinoma cell line model by overexpression and knockdown. Secondly, I performed a multi-omics screen. Quantitative proteomics allows me to identify FAT10-associated proteins, while interactomics allows me to identify FAT10 interacting proteins. Targets that are identified by both these experiments can be inferred to be destabilized or stabilized by FAT10. I also performed metabolomics and lipidomics to identify FAT10-associated organic acids, amino acids, and lipids. I finally validated these omics experiments. In short, my data shows that FAT10 systematically modulates metabolism-related pathways. FAT10 upregulates the glycolysis pathway, and this flux appears to be channeled into lipid metabolism, which is also upregulated. This culminates in triacylglycerol accumulation. Moreover, FAT10 appears to concentrate glycolytic flux by downregulating the pentose phosphate pathway and associated nucleotide metabolism. Interestingly, in addition to reducing the supply of nucleotides, FAT10 also reduces the demand of nucleotides by downregulating nucleotide-dependent processes like cell cycle and transcription. These results suggest that FAT10 is involved in shifting cells from a replication and growth state to that of a survival state by downregulating energy and resource consumption processes and simultaneously increasing energy and resource uptake, production, and storage processes. This FAT10-associated phenotype could help cancer cells survive metabolic stress. Hence, the second aim of my study is to investigate the effect of metabolic stress on FAT10 expression. And I hypothesize that FAT10 is inducible by metabolic stress and it in turn reduces metabolic stress to increase cancer survival. Indeed, my results for the second aim are as hypothesized, and overall I show that FAT10 is an immunometabolic survival factor that is responsive to inflammation and metabolic stress, and which initiates global metabolic changes to relieve this metabolic stress for cancer survival. This is the end of my presentation. Thank you for your time.